So you've heard from both Jim Eubanks and James Ellis about the pathway we're looking for when we're looking with model systems. It's this somewhat idealized form where we begin to understand the genes that are involved from human studies. So it's often the work that you've heard of this morning, work of Dr. Steve Shearer at Sick Kids, for example. And from that, once you've identified a gene of what is happening or what a likely cause for a particular disorder might be, we can create models from that gene. So we could go to the stem cells from that individual, the way Dr. Ellis is describing it, and look at the properties of those cells. Or we could create a mouse, knowing what gene is wrong, and try to find the same gene in the mouse and downregulate it, upregulate it, try to make it to be the same as the human mutation, and then look at the behaviors in the mouse. Eventually, as we understand more and more about the biology, we can begin to target drugs or other treatments towards fixing whatever we identified has gone wrong. We can begin to try to find the same biomarkers between the mice, the cells, and patients, and eventually get to targeted therapy and hopefully successfully improving some of the symptoms that were identified to have been caused by that particular gene. Now, there's tremendous progress and tremendous excitement in this kind of field, but it becomes quite challenging, as one of the questioners alluded to, when we're talking about the complex disorders like autism, ADHD, OCD. This is a bit of a reminder from what you heard this morning. When you're looking at genetics, you can have the monogenetic disorders. So Rett syndrome is one where there's a single gene, in that case MICP2, which causes the disorder. If you have the gene, you have the disorder. It's a fairly straightforward relationship. When we're looking at autism or ADHD or OCD, we're really in the realm where we can only identify about 20 to 30% of the cases with a particular genetic mutation. And they tend to be rare genes that are highly penetrant. So what this means is the individual gene does not occur often in a population of patients, but when it occurs, it has significant impact on that particular patient itself. So to give the example from autism, this can probably be updated a bit, but roughly 70% of identical twins are concordant for the disease, which means if one identical twin has autism, the other one will likely as well. So we know it's quite heritable. We know there's a strong genetic component to it. But when we then look at the genes, we can only identify about 20% of patients, a gene in 20% of patients. And in those cases, no single gene accounts for more than 1% of patients with autism. And the results are not too different if we're starting to look at OCD and ADHD as well. So how do we go about creating a model system for something which is this genetically complex? And effectively, what we're trying to do at this point is to model autism as a collection of these types of rare events, the rare genes, whether it's the ones you've heard about earlier on, like SHANK2 and PATCHD1, and others, and try to see if we look at autism as a collection of all of these different disorders, with each individual having a different potential cause, can we understand more about the disorder through this? So effectively, the way this would work is you create a mutant mouse, so similar to what Dr. Eubanks was describing. We then, of course, given that it's mice, can create more and more and more of them so that we can study a population of mice and really get significant results to figure out what is different, what isn't, what is just idiosyncrasies from one mouse to the other. So we usually take a collection of these mice, so 10 affected mice, 10 unaffected mice, or somewhat more. We study them with some assay. My favorite choice is brain imaging, which I'll talk a little bit more about later on, and then do data analysis on this. So this is the model that Jim Eubanks was describing of, for example, MICP2 deletions or Rett syndrome. We begin to understand what does this one gene do. So we're looking at autism rather than having a single particular gene that's affected. We would look at as many as we can, each of them individually modeling a particular mouse. So if you look at gene one, it might be, let's say, PATCHD1, for example. In the middle, you might have SHANK2. You might have 16p11 copy number variants, 15q13 copy number variants, et cetera, et cetera. Each of them have their own phenotype. Each of them have their own symptoms that they present. So whether it's they have motor coordination problems, whether it's they have social interaction difficulties, it might differ from each case to each case. You heard a bit about this in James Ellis's talk, where he was looking at just the cellular features of PATCHD1 and SHANK2 and found that there was more in one and less in the other. So even in two cases which are known to be involved in autism, you're starting to have different results already. So the question is, what should we measure as we're getting into these different disorders? We can look at behavior in mice, and Jim Eubanks gave you a good overview of it. We can look at reciprocal social interactions. Basically, is a mouse more interested in looking at a toy or looking at another mouse? Is it more likely to remember if this is a mouse that it's already met before or not. We can look at hyperactivity in mice as well. How much do they run around? There's repetitive behaviors. In mice, this often takes the form of repetitively grooming themselves or obsessively burying marbles if they're given marbles in their cage, which is related to restricted interests as well. We can look at some ultrasonic vocalizations. Are they more likely to cry? This is something we can't hear, but with advanced microphones we can pick it up. Are they more likely to cry as a pup if their mom is taken away from them? We can do these kind of measurements and get an idea of how does the mouse behave. But this is one of the dangers in trying to ascertain, is a mouse really like a human? That none of these behaviors, even though they're related to what we see in humans, is really the same thing. Ultimately, a mouse is still a small animal, small squeaky animal with a tail. It's not going to behave exactly the way a child would. So we tried to go to more biological assays in addition to these behaviors. And the assay of choice for us is brain imaging. So what you see here is on the right, you have a patient scan from a 6-year-old, I believe. And on the left, you have a scan from a mouse. Now, this is not to scale. We do breed mice big, but not quite this big. But it is the same thing that you're doing in the mice that you're doing in the humans. You can begin to get some of the same assays, and then you can use that to try to map between some of the cellular things you can see, as you heard about earlier, some of the behaviors, and the behaviors we see when we're looking at patients as well. So I'll give a few examples from autism, because that's where we have the most detail. We've been trying to collect as many mouse models as we can. So here's a list of the ones we've studied so far. These are 26 different mouse models. And each of them corresponds to an identified genetic variant known to be involved in autism in human populations. Or in 2 cases, a mouse which is behaviorally autistic, so less likely to interact socially, more likely to have repetitive behaviors. So effectively, rather than studying each of these individually, which is an important work in science as well, trying to understand exactly what each gene does, we can ask how does this collection of all these different individual mutations, which is what you normally would see if you were to get 100 patients from the population or from any of the studies we do, about 20 of these patients would have some of these mutations listed here, but we'd like to only have a single patient with associated with a single genetic mutation. So then we can look at, once we've acquired the brain images, once we've looked at the behavior, we can look how similar, how different are all these different mutations to each other. That's what this plot over here shows. So on the right-hand side in the blue, you have the total brain volume, and on the y-axis, you have a measurement of how different is it from otherwise unaffected similar mice, so often from litter mates, from the brothers or sisters mice who are not affected by that particular mutation. And the measurement is in effect size, or it's in standard deviations. So effectively, one standard deviation, if you're within one standard deviation, you're similar to about 67%, 2/3 of the unaffected litter mates. And as you get away from that, becoming more and more dissimilar. So what's striking about it is, as you go across the different genes, each of them listed on the x-axis on the bottom, is you have some mouse models that result in brain undergrowth, smaller brains. Some mouse models result in brain overgrowth, larger brains. And that's a consistent type of feature that we see in autism. Once you get away from the clinical diagnostic criteria, if you hear researchers say that autism is something, odds are they're only talking about a small segment of the population. Autism really is this very heterogeneous contribution from different genes producing different phenotypes or different outcomes, which somehow in the end converge to similar symptoms that have the same diagnosis. In the red on the left, you have the same kind of measurement, so effect size, so how different is it from its siblings. But in this case, you're looking at the hippocampus, one structure in the brain that's really involved in emotional regulation and especially learning and memory. So again, in some mouse models, that is enlarged compared to their siblings. In some mouse models, it's smaller. We can take some of these data and begin to say, well, of course, everything is quite different from one model to the other. Is there anything that begins to be shared across all these different mouse lines? So we can ask what areas are consistently implicated. So the 2 different rows each show the same kind of idea, where you're looking at how likely is a particular part of the brain to be affected by each of these 26 mutations. And so the brighter the color, the more likely it is that every mutation has something related to something happening in that area. And you see that it's a few areas that do come up as somewhat consistent. So you have especially the cerebellum of the brain, which is fascinating, because if you were to study an older neurology textbook, you think the cerebellum is really only involved in motor controls and getting feedback. So the classic cerebellar test is if you were to touch your nose, we can do this in a smooth motion. With a cerebellar lesion, you have a hard time adjusting and being able to touch your nose in one go. not something we classically associate with autism, but it seems to have some role in just feedback and feedback regulation across more than just these kind of motor skills like touching your nose. There's areas in the cerebral cortex involved in higher order cognition. There's the striatum and the hypothalamus, one in motor, one in more emotional and autonomic regulation. So areas that come up again and again do seem to have something that we know about the disorder, and they are in common across all these different individual mutations, even if you're not guaranteed with any one mutation to have something showing up in those parts of the brain. But of course, the nice thing about brain imaging is that it is the same measurements we're taking in the mouse as we are in humans. So if we look at our patient population, we can do the exact same kind of measurements. So you again have on the y-axis brain volume. How different is a particular child? So in this case, each bar represents a particular child in our studies. How different is it from the closest typically developing children that we also scanned? So for each child, we tried to find 15 controls that were approximately the same age, that were of the same sex. So boys match to boys, girls match to girls. An 11-year-old matches to another 11-year-old, et cetera, and say, how different is it? And we see this widespread again. Most of the kids will have no difference in brain volume from their controls, from other non-affected kids of the same age and gender. Some will have smaller brains. Some will have significantly larger brains. So if I bring the mouse data back up, you see something that looks remarkably similar. You see this wide spread of some larger, some smaller idiosyncrasies across it. And again, just like with the mouse, we can look at not just the whole brain, we can look at the hippocampus, and you again have the sum up, sum down. So autism really is a widely heterogeneous, a lot of variability in the disorder. And that's recapitulated if we look at these individual mouse models and combine them together and treat them as our autistic mouse model population, i.e. a lot of different mice, each with individual mutations associated with the disorder. So where we're hoping to take some of this data is to start looking at clinical trials. You heard them alluded to before from both James and Jim. But the idea is that normally you would study in preclinical trials the idea of, can you cure a particular syndromic form of a disorder? So can you cure MECP2 deficient mice which model Rett syndrome? Our question is to take this to the complex disorders like autism, ADHD, and OCD and ask, can we infer more about each individual and each of these mouse models to understand who is and who is not responding well to a particular drug? Who does the drug help? Who doesn't it help? Who has some adverse effects? Who does not? And I'll finish with a study which really comes from the cancer field, which has inspired us to do this work, where they had some of the exact same effects. So in this case, they were looking at lung cancer. And what they had is they had a particular drug. In this case, it was a MEK inhibitor, or silumetinib. And they found that only in about 10% of the patients did it improve their outcome for lung cancer. And they wanted to understand, why is it? What do we know about the 10% that are helped by the drug that separates them from the ones that don't? So they had some hints, but it wasn't very clear from looking at the phenotype. How bad was the cancer? How advanced was it? Was it in an older subject, in a younger subject? And so what they then did is they started to take mouse models and implant the same lung cancer in these mice. And the mice had different genetic backgrounds. And what they found, there was one particular gene, in this case it was LKB1, which if you had that gene, the drug did not help at all. If you didn't, if you had some of these other mutations, in this case CRAS and P53, the drug was 100% helpful. So the ability to use the combined information from the mouse with ongoing clinical trials to figure out who is responding and who is not, and get to a more individualized treatment is where we hope to end up with this work going from mouse to humans to trials. So of course, many, many people help with this kind of work, and thank you for your attention, and I hope you enjoy lunch.